Good morning, everybody, and thanks, Professor Abramovich, to introduce me. What I will do this morning is give you an overview of retinal image analysis. It's a relatively new field. We have a look at MRI, CT, etc., and now we have discovered also images from the retina. It's actually images from the brain. And I go talk a little bit about brain-inspired image analysis. The best computer we know for pattern recognition is our brain. So what do we know today of brain studies? This whole field is part of medical image computing. It's a huge field in the world. People do computer-aided diagnosis. We have enhancement, visualization. I have a group in Eindhoven University. I'm now retired, but this group was always have a successor, about 25 people. And we worked very closely with Philips Medical Systems, our neighbor, 4 kilometers away. So we have dozens and dozens of students, collaborations with them. It's a very nice and close, intense collaboration. And our main field is computer-aided diagnosis. As you know, we make images by the millions. We need more and more to be precise, to be quantitative. So the classical pipeline is that you have some pre-processing, denoising, you segment what you like to see, like blood vessels, tumors. You want to find out what type of tumor is, so you do feature extraction, and then you do feature selection. You have some classifiers, support vector machines, and then you say, okay, is it healthy or is it disease? And you mark the spots that you find as a computer, this is suspect. The classical machine learning approach designs all kinds of filters. You make a feature space, and in this feature space you get clouds, and then you validate, you say, okay, this red is wrong, this is good, and you have all kinds of techniques to find out, these are receiver obturating curves. How good is this technique? And the way we today work is, especially with MRI, we make images like this in about 5 minutes, completely 3D. And these are then 512 by 512 by 512. But CT scans are even more productive in terms of, I have a small movie here. Let me see. This is a multi-slice CT scanner. It generates 250 slices per rotation. It rotates 3 times per second. So this machine makes 750 slices per second. So to make thousands of images of a lung or a whole body is extremely easy. It should stop. You see the detector here, and this is now 1,000 detectors. So we make images 1,000 square with this incredible volume. And which doctor is going to look at 6,000 slices of the lung? We need computers to find these things. MRI is now generating 4D data. That means you have a cube of the MRI, of the heart, for example, 512, 512 by 512 to the third. But this cube is measured 25 times per second. So that's a huge data volume. This is the aortic arch, and in my group we did a lot of visualization. And what you see here is 4D MRI. And we visualized for the first time the flow in the aortic arch. And we could now see that there was turbulence here. And this is now routinely studied to see if the valves are closing very well. This could not be seen before. There are lots of kids with what's called aortic coarctation. This doesn't have any radiation. There is no X-ray. There is no catheter inside. There is no contrast media. It is just a 5-minute MRI scan, so quite special. We have done this with Philips. If we go to cancer detection, we use everything that you can use from these, not only the anatomy, but also the function. If you have normal blood vessels, it looks like this. But if you have blood vessels in a tumor, they don't have enough oxygen, so the tumor is going to do what's called angiogenesis. It generates new vessels. But these vessels are very sloppy. They're made way too fast. They're leaky. So in the tumor, you have different blood vessels, and you can see them by measuring a time series and then measure the flow. And if the flow is very fast, so here we have the uptake of the contrast medium over time. If it is really a fast washout, it's malignant. If it's very slow, it's benign. So you can mark now the pixels. Every pixel you can see how fast was it actually flowing away. And then what you do is you mark all this. But I go to deep learning because that's now artificial intelligence taking over all these classical feature approaches. And with deep learning, we have now fantastic results in finding, actually currently better than human performance, the detection of all kinds of malignancy in these images. And I go talk a lot about deep learning. And why is it called deep? Because it has many layers in the neural nets. This is the famous AlexNet. You put in the beginning your image, and you have a lot of filters, in this case 11 by 11 pixels. You convolve this in the image, so you get a new image. But because I have many filters, I also get many output filters, so in this case I have 48 filters. So you get a tensor here. Well, this tensor is convolved again and again and again. So these many layers means I have multiple, this means it's a deep neural network. And at the end you have a fully connected layer, and this is a classifying layer, and you decide what is it. If you do digit detection and you put in handwritten digits, you have 10 classes, 0 to 9. If you have a disease, you can have, let's say, healthy or benign. And this very much looks like the multiple stages we see in vision. After the retina, it goes to the center of the eye, and here it goes to the visual cortex. And the visual cortex is huge. 1/4 of your brain is for vision. That's amazing. We are really visual machines. That means we have hundreds of millions of filters, and we do exactly this. We have all these filters here. In our modern neural networks, we easily have, let's say, lots of stages, and we also have hundreds of millions of filters here. So I'm going to talk a little bit what happens here. In the retina, we have these cells that project to the first visual cortex, V1, different layers. Then it goes to V2, V3, V4, and there are, well, at least about 15 of these layers now recognized, and there's tremendous research going on in the biology of these vessels, of these layers. And I'm going to talk a little bit about this. I found out that these worlds of biology and computer science are quite separate. Biologists never use mathematics. They have a lot of knowledge about cells and pharmacology and so on. And in the computer vision world, people don't know about all these cells. People have never heard about stellate amacrine cells in the retina. You have 100 million of these cells. They do histogram of gradient detection. And most of us, we just don't know. And there are fantastic means to the revolution in computer science. The revolution in biological science is just as big. We have optical methods, and I'm going to talk a little bit about these things. The reason I do this is for retinal image analysis, so I start my overview a little bit with what do we do with the retina? Why do we do this? Then I go a little bit deeper in deep learning, and the last part is it's a black box, this whole deep learning. But learning so much now from the brain, can we also try to understand what is this black box actually doing? What is the self-organizational principles that we see in the brain? And you will see that's quite striking. We have discovered a lot recently. The reason we do retinal image analysis is that diabetes is exploding in the world. And we were asked by China, our university, to help, because in China it's particularly a big problem. 11.6% in China has diabetes. And why is diabetes so problematic? Your blood vessels begin to leak everywhere. And the bleeding spreads out to your kidneys, to everywhere. And you can have years that you don't see it or don't mention it. But it turns out that the retina is a very special tissue. It uses so much oxygen, it actually uses the most oxygen of your whole body. It even has two blood flows, two networks, on top and on the bottom. So if something is compromised with your blood vessels, your retina is one of the first to damage. And you get blind. So 4% of all the diabetes people, and we have hundreds of millions of them, they start to see these spots. And nerve cells never regenerate, so this will be forever. And if you start to see this, you're far too late. So what we need to do is to find the very, very early signs of beginning damage to these blood vessels, small bleedings, small changes in the vascular with the vascular curvature, and that's exactly what we do. And in China, that's a huge country. Actually, it started when Philips went to China. They said, well, we want to sell our CT and MRI scans. And they made a joint venture with the biggest medical imaging company in China, 20,000 people in northeast China, Shenyang. They needed new engineers, so they started a new school. And they signed a 20-year contract with my university. This school is now mature. It's now established for, 9 years. They have 1,000 students. And my boards came to me and they said, Bart, you are our imaging professors. These guys are going to do imaging. Can you advise them? Can you set up the whole research team, appoint new professors? I went there and it was amazing. It was fascinating. It was really a renaissance, all building up. That school was founded, and then a major hospital group of eye hospitals came to us and they said, well, we need your help. We are going to set up a screening program for the elder half of the population of this province. That's 48 million people. We're going to scan 24 million people, cameras like this, we make these kind of images, and we need computer software to do this automatically. By far, we don't have enough doctors to do that. And today, we have already hundreds of cameras now operational, and I'm going to talk to you about this project. I'm the project leader of this project. It's called Retina Check. We had lots of PhD students. Actually, I recently had 4 PhD students finished on this project. And here you see the big issue. Asia has a lot of diabetes. Why is that? First of all, it's in the genetics. And for China, the lifestyle change is way too fast. 15 years ago, there was no sugar, just rice and chicken and so on, very modest. And now they have all the luxury. It's too fast. And it's very, very luxury. So in 1980, there was no diabetes. And in now, 2018, it is here over 11%. It means 1 in 9 people has diabetes. It's staggering. So there's a big alarm about this epidemic by the Chinese government. And it is still rising. There is no flattening off yet. So it is really frightening. So we set up a Sino-Dutch collaboration. TUE, my university, was designing all kinds of algorithms. I'll talk to you more about it. The Northeastern University did the data analysis. They also made the complete e-cloud system. And we have Maastricht University Eye Clinic in the Netherlands. They do the biggest diabetes study in the world. It costs 30 million euros. It's funded by our government. And they take 5,000 healthy, 5,000 diabetes patients, and they scan everything. MRI scans, Cooper tests, lifestyle interviews, you name it, full ophthalmology room. And they do a full phenotyping study on diabetes. And we collaborate with the biggest hospital in Shenyang, 3,000 beds, to do a field test with scanning diabetes patients. So what do? We focused primarily on the vascular network. So we looked at vessel integrity, leakage, and all these tiny things. I'm not going to talk about all these tiny details, but we can detect 17 biomarkers in these images. Early bleedings, early changes in vessel width, etc. So the trick is that people can see this too, but people cannot see how much are the vessels curved. You have no number, we cannot estimate that. And computers can very accurately do quantification. They can count all these microbleeds. They can see the progression of the disease. Is the treatment really working? So the other special thing is that retinal vessels are brain vessels. You directly look at the microvasculature of the brain, and this is far cheaper than MRI. People also make MRI scans to study microbleeds, etc. But I can, for no costs, it's just a regular camera, I take a 12 megapixel image with 10 micron resolution, which is far better than MRI. That's half a millimeter. I talk about 500 micron, and far more expensive. So we have a very inexpensive way of looking at the brain, and currently we're looking at all kinds of neurodegenerative diseases, like Alzheimer. they also have very early signs in the vasculature. And people cannot see it, but artificial intelligence can see it. So we have a whole new modality coming up, which is spreading quickly. So we do vessel tracking, segmentation, enhancement, microaneurysm detection, curvature, we also call that tortuosity. So what is, if vessels get weak, and the problem is the vessel wall, if you have these very high glucose content, the vessel wall breaks down. In brain vessels you have what's called the blood-brain barrier. The brain is very well protected from all kinds of chemicals to go in. And that means that the cells in these vessels have very special junctions, and these junctions break down. And you can actually see that. But when these vessels break down, they also start to be more sloppy. They get wider, they get more curved. And we can see this very quickly. We study the fractal dimension. It's actually the filling of space degree. So if there are new vessels growing, we can quickly see it. We find landmarks to do like bifurcations and crossings, and you can easily then do panoramic stitching and find a larger area. We measure bifurcation angles. Normally vessels, they go straight, directly to the place where they have to bring the oxygen and the sugars and so on. But if the vessels are diseased, they go among all kinds of other angles. So all these are what we call biomarkers. And because we have all these biomarkers, we found that diabetes is related to dose. Glaucoma, high eye pressure, is another disease, very, very often occurring, is related to dose. Hypertension, to dose. So actually what we said is, hey, we can make a one-stop shop. If you measure all these parameters, you can just give a probability. You say, hey, you have a very high probability of having diabetic retinopathy, disease in your retina. But there's also some chance that you have glaucoma. So we measure now actually all these diseases by measuring this full set of qualitative parameters. So it was a whole setup. You see me signing here the contract with Professor Ge. He's the leader of the conglomerate of eye hospitals. That's in total 1,200 people, 23 hospitals. It's huge. He has a very big access to the Chinese healthcare market. For us, it was fantastic to know a guy like him. So we became very close partners and friends. In 2 weeks, he will visit Eindhoven again. This is the Minister of Healthcare. This is the President of the Academy of Science in the Netherlands. And this is from the Technical Academy. Here you see me talking to the Minister of Education. This is the ambassador in China. And this is the CEO of a company that makes very innovative laser scanning cameras that joined our consortium. But I want to go to the challenge, find disease, and this is a very nice image that illustrates the difficulty for computer vision people. The artist here made faces, and for us it's very easy to see a face here. And you see a face here. But now you have to write a computer program to detect these faces. So I asked my students, well, go ahead, do this. And of course people now say, okay, we can train a neural network and you learn at lots of faces. Face databases are easy, you can easily get millions of faces. It's not that easy at all. And this is actually what a doctor needs to do when he finds the tumor. He needs to scan this image. And when you help him, you say, hey doctor, our computer vision algorithms have now find the spots, hey, look here, look here. And now you see, you didn't see this before, but now you see it. This is very difficult to see. So it really helps. And doctors are extremely happy with this help in computer-aided diagnosis. So, of course, you have also now, it's getting too good, better than the humans, so there's also worry that they say, okay, are we losing our jobs? And there's something really seriously going on. Yes, artificial intelligence is getting so good that they have to take care on how is this involving our radiology profession. They own a lot of data, and all the big companies, Amazon, Google, Baidu, they are now more and more entering the hospitals. And there's only one reason, give me your data. These hospitals have PACS, picture archiving and communication systems. Millions of these images are there, and they want to have these data because a human expert can learn, I don't know, maybe 10,000 patients in his life, in his career. Good one, maybe 15,000. but these systems can learn 50,000 patients in a week. So that's quite staggering. So actually what I like to focus a little bit on is how do we do it, and then go to brain-inspired computing. So I'll tell a little bit about imaging the brain. We know all about MRI anatomy. This is my own brain. You see my spinal cord, the whole thing. This is wonderful. It has a superb resolution, but it's actually only anatomy. And you all know that MRI is very versatile. With MRI, we can measure much more. We have today 26 different physical measurements with MRI. We can measure the flow, we can measure the local pH per pixel, we can measure the local oxygen saturation, we can measure the spectrum, so how much hydrogen, we can measure the flow, lots of things. This is another MRI technique. If you use part of your brain, suppose I use my hand, then I use this motor strip. Here is all my muscles here, and somewhere here is my hand. So this is active. These neurons, they give action potentials. They need oxygen, so they give a signal to the blood vessels, please bring more blood, open. And blood has oxygen, and oxygen is paramagnetic, and you can see it with an MRI scan. So you can actually see active areas with MRI. It's called functional MRI. And you see here with visual stimulation that the whole back of the brain is now visually stimulated. And this is a huge field in which people can now very carefully map activities with locations on the brain. It has really high precision, and most studies are now done with 7 Tesla MRIs. You have voltage-sensitive dyes. That's actually, I'm not sure if the movie is working. It's a pasta you put on the brain. You do this only on experimental animals. And when these cells give an action potential, they give a very light blue light flash. So you can actually see single cells firing. And under a microscope, you can see whole patterns of firing, real time. Well, this is spectacular. They used to stick a needle in one of these cells, and you could stick 10 or maybe 100 needles, but you saw almost nothing. You saw a few letters of the page. But now we can see the whole page. We can see the whole story in real time. So voltage-sensitive dyes was a spectacular breakthrough. A recent kid on the block is what's called optogenetics. This is the molecule that works in your eye, in all your rods and your cones. That's light sensitive. It's called rhodopsin. It is a molecule that goes 7 times up and down. It is located in the membranes of your rods and cones. And if a photon comes in, this molecule opens a little bit, and there's a little ion flux that gives a small signal, and that gives then the light sensitivity. But they found the DNA of this molecule, put it in a virus, and injected the virus in the mouse brain. And the virus said, okay, my DNA should go into these cells, so inject the DNA in these brain cells. These brain cells started to make rhodopsin, and you get light-sensitive brain cells. And now you can stimulate brain cells 1 by 1 or 100 by 100 by visual stimulation. And they make blue light to open the gates, so excite the neurons, and yellow light to close the gates, so inhibits the neurons. So this is a small movie of a mouse, and it has a light-sensitive fiber optics. Now the light is on. It's located on the motor cortex. And you see it stimulates the motor program. And when they turn off the light, the mouse stops. So it's quite amazing how you can do very careful studies by stimulating, by measuring. And another one is connectivity in the brain. And my group has been very active on this. We can now measure the wires in the brain since about, what is it? I think about 7 years. The technique is as follows. Water makes Brownian motions. It's constantly bumping to other molecules. And actually you can measure this diffusion process, this Brownian motion, with an MRI scanner, because if you make a high gradient, so you have a 3 Tesla scanner, and here it is 3.1 and here 2.9, if these molecules move a little bit in this direction, they change, even if it is nanometers, they change a small magnetic field change, and they change frequency. These frequencies can be measured. So if you do water in an axon, a neuron fiber, it is restricted, and it can much easier move in this way than in that way. So if you measure for some time, you find an ellipsoid. Actually, you measure a tensor, a 3 by 3 tensor, and the eigenvectors of this tensor, you do some linear algebra, give you the 3 main axes, and here you see the ellipsoid, and you see there is already a connection here. And when you do this in all these millions of voxels, you ask the computer to do the connection. In water it is isotropic, but here it is anisotropic, and then you can measure beautifully all these connections. And in my group we wrote a lot of software, all in CUDA on a GPU, a graphical processing unit, to visualize real-time all these connections. And here you see the connections in the brain. In one plane I show you all these ellipsoids, and this is the corpus callosum, the connection between left and right brain. We used color coding for the orientation, so X, Y, Z is red, green, blue. And we used also shading and interreflections by studying the physics of hair. So if you have a horse in the sun, you easily see the directions of the fibers by the specularities. And on a GPU, you can easily add them. So we used that in finding, this was a patient that was operated on the brain. But here are also the optic fibers from the eye to the back. And some of these patients lost their vision because they went too deep. So what we needed to do is find these tractography. It was noisy. We removed the noise. And now we could finally see this is the optic tract. And in this patient, you only needed to operate 24 millimeter deep. So we could nicely visualize the optic radiation tracts. And this patient was saved from blindness. And this is now routinely used. So that's a very nice technique of, inside the brain. So we know the connectivity, we know the function. And if you stick a needle in V1, you have this in the back of the brain. What do you see that this cell is actually seeing? Well, it turns out that this cell is connected to a whole set of cells in the retina, and it's called a receptive field. And this receptive field has a positive and a negative area. And it is looking like this. We have 50% positive and a negative surround, and 50% the other way around, negative here. So why is that? And if you go, this is in the retina, this is in the middle of the, this is in V1. And in V1, we started to see the first filters. And these filters turned out to be derivative filters. It was quite amazing what was found. This was found by Hubel and Wiesel. They got a Nobel Prize for it. And we did a lot of modeling on this because the type of cells that you find in V1 are exactly the derivative operators. This is the first derivative in X and in Y, the second derivative. And these are derivative of Gaussians, because if you look at the mathematics behind it, if you do an observation, you use an aperture, you use a pixel. And actually the pixel is in your eye, and that's a Gaussian pixel. A Gaussian pixel is nice because it has no artificial edges and so on, no squares. And you do a convolution, but if you want to have the derivative of this observation, you can move this derivative operator inside. So actually you can also convolve with the derivative of your aperture. And the derivative of your aperture, here you have a whole set, and this is exactly the set that is measured in V1. It is also the set that people are using in deep learning. This is exactly the set that you get when you do backpropagation in a deep learning network. You find these cells in the beginning. So we see already a beginning with the first cells. But of course, this is very local, and we need to go to global operations. For people, it's very easy to see that these are connected lines. Computers see lots of lines, and you need to have a more global view on this. So we started to look in the literature, and this is a voltage-sensitive dye measurements of the brain. It was done in a mouse, when they show lines at different orientation. What a mess. But it doesn't be a mess actually at all if you look very carefully. And we visited this lab. You see here the place where the mouse is located. They have all kinds of cameras and very high resolution, real time. They can see this activity. And when you zoom in, this is a small area of the cortex, orientation of the line, you see that the orientation is located to regions, and these are called cortical columns. And people have very carefully studied these cortical columns. A cortical column is about 250,000 wide. You have thousands of these columns. And it turns out that this column is the full processor for one pixel in your visual field. So you have hundreds of millions of cells who are working to process your single pixel. And these columns are also connected. So this is a column. And on a column, it turns out, all these cells, they are very nicely organized. So they have different orientations in a pinwheel structure. Here you see the different orientations. In the middle is a singularity. This is the biggest receptive field. There's also different sizes. So we have very tiny ones and very big ones. So it's multi-scale, multi-orientation. The tiny ones are at the boundaries, and the biggest one is here. And here you have this, all these singularities, how they are located. People also started to study, hey, you see a pinwheel here with a singularity, all these different pinwheels. They injected some ink here in a blue area, and this blue area was a vertical detector. And they waited for several days, and they saw that there was a diffusion of this biocytin to other areas. And all these areas turned out to be blue areas. So this guy is talking to all the blue areas in his neighborhood, and it turns out to be an elliptical area. So that means if I'm a vertical detector, all my other neighbors, they tell me, hey guy, I'm also vertical. And those on top, they also tell me I'm vertical. So the probability that I'm vertical by this voting process makes me a very robust detector. So this is contextual information by voting process. And we do this all the time in all these different orientations. So that's a very nice thing to do. And of course, we modeled this in mathematics. So we said we have this orientation, and we looked at the Fourier domain. Here are all the orientations. So okay, let's take one wedge. This is a set of orientations. Here we have all these wedges. And we looked at filters that combined all these wedges. So all these wedges together, they're perfectly fine because they don't throw away information. If you take Gabor filters, they do throw away information. They only have a very tiny spot of this. So you need multiple Gabor filters to have at multiple scales. It's a lot of work and a lot of extra work. And our filter was far more effective because you have all scales at once. And if you have, let's say here you have about, 24 of these filters, you can you filter this image by this rotating filter, and then you get a new space. We call this the orientation space. And this is actually measured in these pinwheels. And this is a very rich structure, and we did differential geometry on this structure. You can do all kinds of measurements on this structure. What is so nice here that in one pixel, you have, this is x, y, and this is theta. In one pixel, you can represent multiple representations of multiple orientations. So here you have multiple orientations, and it was never clear what to do. But now we have separated this because it's all in different fields. So we have expanded our orientation space. And this turns out to be very nice in, this is tissue from our tissue engineering department, new heart valve tissue. And they had troubles. This was a laser scan, it was noisy. It needed to be quantified how many vessels are, how many fibers are parallel, because then the tissue gets stronger. It was never possible to really denoise this because of all the crossings, but with our expanding, extracting these orientations, we could now perfectly denoise these systems. So we had a lot of effort from that. We used this in all kinds of vessel tracking techniques, and we solved actually all the different problems from what happens at crossings. You see there's a crossing here, and by our multi-orientation technique we could nicely do this, even if they're very close together. If they go from very thin to very thick, we could nicely do all this. This is very slowly analysis, but we can do now in 1 millisecond a full multi-scale and multi-orientation analysis of the retina. We also developed techniques for automatic microaneurysm detection, and here you see the result. The brightness of the ring is actually indicating the probability that we can assigned to the certainty that we find it. Okay, a little bit about the pipeline. Images are often not homogeneous. Could be due to the camera, due to the illumination. So you first ask the computer to make it fully homogeneous. This is much easier for inputs to the next stages. And here we have a very nice application of our multi-orientation filters. We made these filters half filters, so they only point in one way. And if you stand on a pixel, and suppose you stand on a blood vessel, in this direction you have a lot of output because you measure on the blood vessel. Here you measure next to the blood vessel, there's no output. You turn around, high output again. So on a vessel I get 2 maxima, but on a bifurcation I get 3 maxima, and on a crossing I get 4 maxima. It's very easily calculated. So with these filters we could now, in a few milliseconds, we have all these marking points where we say, okay, here we have all the bifurcations, here we have all the crossings, and then we studied carefully all the different angles of bifurcations, and we found that in diabetes patients, these angles were markedly different from healthy patients. We used them to stitch these images, and here we see a measure of tortuosity. And how do you measure tortuosity? Well, tortuosity is curvature, and we know the formulas of curvature, of course, and every vessel center line has a different curvature. For example, this has no curvature, it's straight. This has a positive curvature, it's concave. This has a negative curvature, it's convex. So we have thousands and thousands of curvature measures here from this image. So what we actually did is we measured the histogram, and if the histogram was very narrow, it means all the curvatures were 0, it's a healthy patient. And if there was a lot of curvature here, indicated in color, we get a wider histogram. And you see here that with healthy patients, you have a low curvature, diabetic retinopathy, a higher. And we did extensive studies and validations, and curvature is an extremely early biomarker for diabetes. And that's now extensively used. We were one of the first to indicate that. It's difficult to measure this curvature, and what we actually did is in this orientation space, we had this wonderful multi-orientation space, we fitted circles, so we get these very intricate structures, and this was highly sensitive, and we could now nicely measure for any vessel, white and not white. So we made a whole workstation, patient input. You can design a whole pipeline here with everything you'd like to do, and it is an automatic report generator. It was designed by my postdoc, and it's now in actual use. But let's go to the core business. How is all this working? Let's go a little bit to deep learning. And you all know about deep learning because it is now in every computer science lecture set. The basic trick is a set of neural networks. There's 1, 2, 3. You have an input layer, hidden layers in the middle, and an output layer. And what is the first layer doing? It actually measures very simple filters. And most people take Gabor filters. It's very wrong, but everybody takes Gabor filters because previous people took Gabor filters. But if you look at the reasoning for Gabor filters, there is no good reasoning for Gabor filters. I think the very good reasoning is this is differential structure. We look at geometry. If I look at the local structure, I make a Taylor expansion, and I can see, okay, what is my local expansion? I need derivatives. I'm a physicist by nature and by training. So we do local differential structure, and then we get long feature vectors of all this high-order differential structure, and these feature vectors we're going to contextual combine in a larger context, in an even larger context. So if I do phase detection, I have first edges, and I want to find the nose, for example, so I have an edge here, and an edge here. Then I need to combine these 3 edges in the next contextual level. That happens here. So here I make them into more contextual things. I make these triangles and eyes and noses. And in the next contextual level, I make this bigger, and I group them into faces. But how can we understand this let's say, discrete contextual increment from this feature analysis? And we do a lot of differential geometry and linear algebra now on finding this. And I recently had a patent on understanding this step. These are all convolutional neural networks because neurons are perfect for convolutions. They are convolvers by nature. We call it the receptive field. That's the filter from a neural network. And as I showed you before, we see exactly the same in the visual field. And the performance is so good that it is now kicking out all the other historical computer vision. So we do not only have the radiologist in crisis, oh help, we lose our jobs. We also have many of us, computer vision scientists. My goodness, all my 20 years of work on feature detection, it's now overruled. It's done by just take the data, download TensorFlow, put it in, it works, and it's far better than I did the last 20 years. So I cannot publish it anymore. That's really true. So you have to go to deep learning because it's better, and the whole world is asking for it. So let's look a little bit in the biology further. We have a lot of layers. It's incredibly detailed. I can only give you a little bit of, but we have actually, these are the retina M cells for motion, P cells for shape. It turns out you have 26 of these P cells measuring in the retina. We measure far more than just intensities. Then it goes to V1. Oh, this is in the middle of the brain. It's called here the thalamus. And then it goes to this big area, V1, V2, V3. And all these are neurons. And neurons are actually quite simply modeled. This is already done for decades. You have inputs, you sum, you have a thresholding operation, and this is the very basic model of a neuron. And that's what actually happens. These inputs are called synapses, and every neuron is connecting to about 50,000 other synapses. These synapses work electrochemically. It takes about less than a millisecond. And then you put them in a neural network. And these networks we had for decades, a 3-layer network, but actually they didn't work so well. It was only 75%. And then we get this ImageNet large-scale visual recognition competition, 1.2 million images, 1,000 classes. Up till 2010, every year there was a game, a challenge to be the best in the world. And then there was a guy, and today we have 96% correct. It's better than humans even. And that made a huge jump in all this. And I told you before, we do edge detection, combine these, combine these, combine these, and then finally we have this whole thing. So the big things that we need is to train this network, you do error detection at the end, and then you say, okay, suppose you have letter detection or digit detection. I put in a 6, but I measure a 7. The error is 1. You adjust all the connections here with nice optimization techniques. You have all kinds of gradient descent techniques. And sometimes you have to update 100 million of these connections. So you need huge number of images. Well, we have these images, medical too, we have big data. And why is retina so interesting? Google is now going into the medical field for the first time. What do they choose? Retina. Because I can easily have a couple of thousand MRI scans or CT scans, but it's even easier to have 100,000 retina scans or even 1 million. In a few years, I have 24 million retina scans. Well, that's extraordinary information. So that's another thing that you need. We have, of course, now the very tiny computers, the GPUs. These cost nothing because the game world is playing them, so these are just game computers. I just bought a whole box with 10 of these NVIDIA Titan XP. They cost 1,200 euros, so I have 10 of these. But each one has 3,600 small computers on board. So that means I have 36,000 computers for 12,000 euros. That's nothing. So this is a supercomputer. And that's another reason why AI is exploding. We can do now brain mimicking by this very cheap hardware. Okay. This is the machine that I bought. It's just a box with a whole thing of these. We program a lot in CUDA. That's the NVIDIA C++ based programming environment. They have, each of these has a lot of local memory. So it's wonderful. And today we know all these applications. The self-driving car is finally coming up because it can see not only pedestrians, and it's quite interesting. Google has Google Street View. They took several millions of streets. And if Google now sees a building, it doesn't know, okay, that's a building. No, it knows it is that building. It is that building on that corner in Poznan. It's quite amazing. So we have far more than just a GPS and seeing the lines on the road. We know really the environment, and we can store these environments. It is stored. So that is why Google makes Google Street View. It is not all these things are for free. Google Photos is for free. You can upload your photos. But Google is, of course, using all your photos to train your network. So it is really the biggest wins. The winner takes all. So if you start working on images and so on, you're actually already far too late, because these big guys, they do the real thing. That's why Amazon, Uber, they all go now in this area, data, data, data collection. They give everything for free to get your data. I worked with a big company in the Netherlands. As you know, we produce about 40% of all the flowers in the world. And this is a huge auction hall. And the whole day, these pallets go on here, and farmers are here offering the pallets. Here you have the guys who buy the flowers from Germany, wherever. But it was not clear if the farmer had the right rose on its. We make 9 billion roses every year, 450 different roses. And we built, with our medical knowledge, an AI system to recognize which rose is it. So we could check that the farmer had not the cheap rose and selling it for an expensive rose, so we could have a check. Kaggle is a Californian company that makes challenges. And they harvest money from companies, and every year they have some interesting challenges. And in 2015, there was the Diabetic Retinopathy Challenge. They had a $100,000 prize, $100,000 prize, and 661 teams were joining this. They all wanted this to have this prize. We also joined, and we ended up to be 17 from this whole range. So that's not too bad. We were in the best 2%. Not the winners, but this was a very big effort of the whole field. The software of the winners was given for free, so we could see what are these guys doing, what are they learning, what kind of network do they use UNets or guns, what kind of augmentation do they use. We learned a lot. And today we have a bewildering amount of neural networks. You call it the Neural Network Zoo. This is an interesting website that explains all these different networks, what are advantages and disadvantages. It also indicates that there's a lot of heuristics. People don't know actually, and you try everything. So most of the computer vision students, what do? They experiment with what every other student is doing. Let's take a little bit more filters. 3x3 filters, 4x4, 5x5. Let's take 26 filters. Now let's take 48 filters. Let's take 17 levels. Let's take 21 levels. It's an incredible heuristics, and everybody's doing the same. It's a little bit waste of time. There's no fundamental insight. So we use all this in this big screening program, as I told you, with 24 million people. I told you this before. I tell you a little bit about the validation. We did this in this big hospital. And in this big hospital, we did this artificial intelligence screening. But we needed validation. Which biomarkers are actually effective, compare different diabetic groups and normals, and to know how much diabetes someone has, you have to record also its state, his blood sugar levels. You have to know how much is he actually progressing in his developing of diabetes. So we recorded in this hospital 25 metadata from all these inpatients, diabetes patients. It was not easy. It's in the northeast of China, in a city called Shenyang. It's big, 12 million people, but I was the only Westerner in this whole city. Nobody speaks English. In the hospital, everything is in Chinese. So I needed Chinese students to collaborate with the nurses, with the doctors. It was incredibly challenging, but fascinating. It was big, and we managed. And one of my PhD students in China wrote a very nice paper on how this metadata were used. So I gave talks to the local doctors with a translator. Here you see the head of the department explaining my PhD students. It was a very nice setup thing. We built a whole room in which we did 30 patients every day. We scanned with a laser scan and a normal scanner. We made it a little bit looking like Dutch with tulips and so on. You see the Dutch and the Chinese flag. And the patients were happy to do that. And so far this should be 2018. We have done more than 3,000 patients right now. Because we connect to 23 eye hospitals, the next phase will be to connect to healthcare centers. There will be 200. We even have rural areas. So this Professor Ge ordered 4 buses, and we have now 4 buses. They can expand. And these are mobile camera buses. They go to a village, and the population can come through. So it's really aiming for... and so far, we did already 60,000 normal people. And how do you get normal people? That's not easy. You cannot ring the door and can I take your picture? Professor Ge has 50 stores where they sell glasses, vision stores. So optometrists, they measure your eye strengths and they adapt a pair of spectacles. And in all these, there was a camera. This is an Italian camera, was positioned. So if people come for a pair of glasses, they also ask, shall I also take a picture of your retina? Huh? Why? Take a picture. And then they explain, you might have diabetes, and we can very easily see if you have the beginning of this damage. Because that's exactly what we like to do. It is an early warning system. So we have in all these systems now this camera, but our final goal, and 4 months ago, okay, I have 3 more slides left. We want to go to, of course, a camera on a smartphone. That should be really nice. We have neural networks to find all these fatty deposits. We can nicely measure this. And I go a little bit at the end, can we understand how things work? And now I go do a little bit of mathematics. I wrote already 10 years ago a very nice study on, suppose you're a monkey and you need to develop your visual system. And it turns out that the connections to your visual system in the beginning are very redundant. Every cell is connected to every other cell. And you make it by seeing. If you take small patches from an image, and this was, for example, a monkey seeing a forest, and you do principal component analysis on these patches, you find exactly these differential operators. It's really nice. And these differential operators, of course, resemble these, the model that we have here. So these are multi-scale derivatives. We do now a lot of Lie group analysis. It's actually a Taylor expansion. But it became even more interesting if you limit your input and you say, okay, I take this as input. So suppose I'm an animal and I have only vertical trees. Vertical derivatives do not exist. They're all 0. I only have horizontal derivatives. So principal component analysis of this gives me the first order derivative, the second, the third, and only here I get the first vertical derivative. So the filters are different if the input is different. And they did a nice experiment. This is an experiment of a cat. Let me click on this.
We regulate the visual environment by keeping kittens in complete darkness from birth, bringing them out for periods of controlled visual experience inside special...
So, in special chambers. So, this kitten has 3 months of his life only seen horizontal stripes. So, when you then ask this kitten to look at the horizontal stripe,
but if the stick is made vertical, the kitten no longer reacts to it.
It has no receptive fields for vertical detection. So what you see is what you learn.
Remember that this kitten saw only horizontal.
So that's a very important lesson. Suppose you close one eye after birth on a baby, and you do this for 3 months, it will never be seeing stereo anymore, because you need 2 eyes to develop your whole stereo setup. So there's a long lesson in the data are your filters. You don't need to design them, you learn them from the data. And that's exactly what deep learning is doing. And biology is doing it. So there is another thing. this is this face detection, and I can show all these faces, but now I show you exactly the same picture upside down. You have never learned faces upside down, because they don't exist in nature. Faces, you always see faces with the mouth below. It's very difficult to see the faces right now, because in your brain is no deep learning system for detection. So we are all deep learning systems, and you see the proof here that it is exactly doing right that. So we did a lot of mathematics on this, and as I told you, we have now recently a very nice patent on, we understand this first layer. It's actually principal component analysis of very rich structure. But if you combine those feature vectors again, and you do some very nice linear algebra, you can find the next layer. And this will probably be repeated. So I expect this patent to be quite influential. I have some conjectures, my last 3 slides. I think the brain is far more intelligent than we know so far. If you look at the retina here, you see a couple of layers here, especially this one, the internuclear layer. It has 2 layers, and all these layers, they measure the positive intensity here, and they measure the negative intensity. You say, hey, why should that be? Why should we measure positive and negative intensity? Because if you add them, you get 0. But if you add them with one delayed, what you actually do is you look at temporal subtraction in vision. And I think the retina is doing temporal subtraction. So we don't send a camera image with pixels, but we only send the differences to our brain. It completely makes sense. This is mind-blowing. This is very new. I just wrote a paper about this. So we see completely different than we all thought we see. But we have so good memory that we keep, so this is highly efficient. I only process here about 5% of the pixels. And this brain is using 25 watts. That's amazing. That's nothing. We have these Google data centers, they use megawatts. So this is extremely efficient. If we learn much more about this efficiency, we can do far more better computer vision. And this may be step number 1. Look at temporal differences. Here you see the differences. We have a retina that is scanning. You constantly scan. And actually, this is a very nice movie from a guy who's doing SLAM. He has a LiDAR, that is an infrared sensing device on his head, and a smartphone. And he's walking around, it's in Berkeley, and he will then explore the buildings around him. So he's measuring, but in his brain, his laptop, he stores the environment, and you see that we don't need to see the whole environment. We just scan it, and we store it, and that means this is a highly efficient way. This can even run on a smartphone. So you can build a 3D internal representation by just scanning, and that's why we have an eye moving with a fovea. It's extremely efficient. When we take pictures, we take megapixels, 25 per second, and we process all these pixels. It's ridiculous. You don't need to do that. You only need to do it once. You take another scene, and you just keep it in memory. That's what the brain is doing. So we also have a very interesting eye. You see this spot here, but you only see one, but there's one here too. There's also one here. And you only can see one. That's strange. And the reason is that your eye is only sharp in the middle. And when you go out, these receptor fields become bigger. So you have very tiny ones here and very large ones here. So we have a multi-scale sampling, and we only see sharp in the middle. That's highly efficient too. And we're now using this in our deep learning networks, that we do the same sampling. We say we have a high resolution here, and we have lower resolution going out. It turns out to be far more effective. You can do with less computing a larger area, and we were better than a human expert. So we already did this more efficient just by looking how's the brain doing it. So we have a lot of things here to do. Okay, now I end with my conclusion. We had all these retinal stimulation. It's very nice to do this for a purpose. The purpose is actually vision, blindness prevention by learning from vision. So that's why I call this vision for vision. So we learn from our visual system. We got a lot of inspiration. We use it to prevent vision loss. So we're part of a big WH program, Vision 2020. I'd like to thank all my Retina members, and this is the end of my presentation. Thank you very much.